I'm curious. I am curious. Curious.
By nature.
Welcome to Curious by Nature. I'm Jessica Johnson from Newswise. We tend to think of the flu as something seasonal, a nuisance we can fend off with a shot and a bit of luck. But what if the real threat isn't just circulating among humans, but quietly evolving in birds? Today's guest has spent his career at the front lines of that invisible border, the place where animal viruses spill over into people. Dr. Richard Webby is a virologist at St. Jude Children's Research Hospital in Memphis, Tennessee, and he studies the influenza viruses most of us rarely think about, the ones still living in aquatic birds, waiting for their chance to jump. In our conversation, he walks us through the intricate, often surprising world of influenza A, including the H5N1 virus making headlines today, and how scientists like him are racing to understand which strains could pose the next big threat. Dr. Webby, welcome. Can you start by introducing yourself and telling us a bit about your role?
Yeah, so I'm Richard Webby. I'm a member of the Department of Host Microbe Interactions at St. Jude Children's Research Hospital in Memphis, Tennessee. What I'm really curious about is what it's going to take for these H5 bird flu viruses to start transmitting from human to human.
I saw an article on Newswise about your research with H5N1 or bird flu, and I was really interested to talk with you more. And so could you tell me, though, first in general, what you do overall?
We're a bunch of virologists, essentially, with a particular focus on influenza viruses. So, of course, the H5N1 avian flu is an influenza virus. Sort of specific research focus is on the viruses that are circulating in animals that sort of pose a zoonotic, meaning an ability to affect humans, or a pandemic threat to humans. So we're really interested in learning more about the properties of influenza viruses in animals and trying to figure out which ones of those are of most threat to humans. So in sort of a more nutshell, what we call this we study influenza at the human-animal interface, so that interface between sort of the source species and humans, and what happens at interface virology and what sort of regulates the movements of viruses between hosts.
I read that influenza A are all bird flus. Is that correct?
Yeah, so we tend to think about flu as being a human disease, which of course it is. But if you go back up the family tree of influenza A viruses from humans or from swine, they all end up sort of in as influenza viruses of particularly aquatic birds of the world. So that's the real natural host of influenza A viruses, is the aquatic birds of the world. And it's from that host that the viruses, sporadically spill out into other hosts. So yeah, it's technically absolutely true. All influenza A viruses at one stage in their life were avian influenza viruses.
Again, I read that there are about 144 strains of these viruses. Is that also correct, of the influenza A?
If you look at the influenza viruses that are circulating in these wild aquatic birds, if you think of a sort of a virus, essentially a circle with these knobs around the outside. Some of those knobs are pretty important for the virus itself, attaching it to host cells to start replication or getting it off at the other end. One of those proteins is called the hemagglutinin, or H, and there are, depending on sort of which study you're looking at, maybe 16 different H types of influenza A within the wild aquatic birds. The other protein is neuraminidase, or N, and there are 9 of those. And the way influenza works, you can get all different combinations. So think of numbers. So an H5N1 simply means you have a virus with an H of type 5 and an N of type 1, but you can get all of the combinations from H1 through 16 and N1 through 9. So that's where those numbers come from, is all of those different potential combinations.
And tell me more about this particular study in Nature about the treatments and antivirals in relation to H5N1.
So some of the things we do here in our group is trying to assess the risk of some of these circulating viruses for humans. An H5 clearly poses a risk to humans. So we look at that from a biologic perspective. Is the virus becoming more infectious for humans? But the other side of that is, what happens if this virus does change and it becomes a human pathogen? What can we do about it? And that's where the particular study you talked about came from. We became very interested in testing our sort of existing arsenals of flu antivirals. We have a couple, trying to see how well would they work against H5, particularly if that H5 was delivered in the context of, say, contaminated milk. We took mice, which are a lab model for flu, and we delivered milk from a contaminated cow into those animals and looked how well our existing antivirals work against it. And then it wasn't overly encouraging, but that was kind of the reason behind it.
What is the risk of death in someone who catches H5N1?
It's a great question. And there are lots of things that influence the answer, and maybe we'll come to that as well. But if you look, what's the data we have now? If we look at WHO website, you'll see, since 2003, there are a little over 1,000 human infections with H5 viruses of, different forms over the years. But the mortality rate is running at about 50%, meaning of those confirmed human cases, there's about 50% of those have ended up being fatal. It's scary on the surface, but we've also got to keep in mind there are probably many, many more cases occurring that aren't being diagnosed. And, how do we know that? We know that because, we've done some of these studies, the US CDC and others have done it as well. So you could go into some of these at-risk populations and look on their blood for evidence that they have antibodies against some of these H5 viruses. And when you do that, you end up that there's, depending on study, in the low single percentages of individuals, at-risk exposed individuals actually have antibodies. So yes, the case fatality, meaning the confirmed cases, the fatality rate there is about 50%. But the true rate is probably far, far less than that.
So also in your study, you looked at the means in which someone was infected. Was it orally, through the eye, through the nose, I guess? And you found that one way was particularly worse?
For that particular study, what we thought when we started that was, well, let's think about the environment that individuals, particularly, perhaps those dairy workers in this case, what are the possible routes of exposure? One, splashing in the eye through contaminated milk. So, they're milking cows, lots of splashing in the eye. So we did some what we call intraocular into the eye inoculations. We also thought, well, there potentially someone could drink a glass of contaminated milk, right? So we did that route as well. And then a more traditional intranasal, like we might have expect from a respiratory influenza infection. Yeah, we did see differences, absolutely. So the route of infection did change the course of disease. In the model we did, they all were very, very severe disease, but there was differences in the timing and how long it took for some of these animals to get sick. And consequently also some differences in how successful the treatments were.
From your study, what I read, or from the news release actually, is from orally was the most intense, or I guess was the highest, the most infection. I don't know how you really describe that, but it was from people who had drinking the milk that was infected. Is that correct?
Yeah. So that actually it was the route that was the hardest to treat. So the least effective model for treatment. We still don't really understand why. I'd like to, but what is it about that delivery of the virus? Is it the range of cells that were exposed, maybe in the oral cavity? But again, we don't really understand why, and it's something we're looking at now a little bit more, is to try and understand some of that route difference.
Oh, okay. So it was harder to treat with the antivirals. They had less effect.
That's right.
What about, were those patients more sick in some ways as well?
And that's, of course, one of the limitations of the study is we have to model these infections somewhere because we do the infections in humans. But and particularly the intranasal versus oral, but the oral caused more of a disease than ocular, meaning into the eye infection. Yeah, maybe that's reflective of what we're seeing in the real world, right? So a lot of the human infections that are being detected in the US now are in dairy workers who are getting conjunctivitis. So probably through splashing of the eye, and it tends to be mild disease. So in some respects, that was good for our model. We also saw that the progression of disease was slower. In the end, they still got severe disease, and we think maybe it's because the virus started replicating in the eye and then had to find its way into more of the respiratory tract and the lung. And when you get lung infection, that's when you start to get the real severe end of the disease spectrum. And so that's what we think. But yeah, the oral route led to very, very rapid and very, very severe disease. Yes.
I read that it also had gone into people's brains. Is that normal for a flu virus to go into people's brains?
The world of flu and neuroinvasion, particularly in humans, it's really unclear. I mean, there have been certainly neurological consequences of flu infections that happens all the time, encephalitis type aspects. But we sort of go more to what we see in in infected animals, particularly with the H5. So finding virus in the brain of infected animals, whether it be birds or the foxes or bears that come along and eat a bird and get infected, finding virus in the brain is not uncommon with these H5N1 versions of avian flu. Most of the flus we get probably don't have the capacity. It's just these really special ones we call highly pathogenic avian influenza. This H5N1 is one of those forms. And these viruses do have a capacity to get into the brain and replicate. We actually have a project going on in the lab now. We have a grad student who's looking at this very aspect because we think the current batch of H5 viruses, meaning the ones with the if anyone's following the number soup, we use the 2344B versions, which again is us unimaginative scientists coming up with names for these groups of viruses. These ones seem to have a heightened ability for neuroinvasion, meaning they really get into the central nervous system and the brain. We think differently, and that's something we're trying to untangle now as well. So again, there's a long-winded way of trying to answer your question. H5 getting into the brain is not unheard of, but we think this current batch might do it a little differently.
And if that is the case, if the virus enters the brain, then that makes it more dangerous or I guess more deadly, possibly?
Yeah, absolutely. Yeah, certainly a lot of the animals naturally infected with H5, mammals, humans aside and cows aside, but the foxes, the bears, the mountain lions, a lot of them have quite severe neurological signs. They have heads turned around, walk in circles. So no, it's actually a pretty horrific outcome. So
Okay.
clearly part of I think what eventually causes the death of some of these animals.
How are cows being infected with H5N1? Do you know if there's like a most common means of spreading the virus to cattle?
Yeah, that's one of, if not the biggest unanswered question we have regarding this whole dairy cattle outbreak is how did it start? We know cows are pretty hard to infect with influenza viruses. It's why it took so long to figure out what was going on in the first place because cows don't get flu. But somewhere, somehow, an avian virus, because again, this H5 is an avian virus, the virus moved from that source into the udder of a cow. And that's what we don't understand. Was it a direct movement? Was it some other bird involved? So we don't have the answer to that. It's clearly, I think, a pretty rare event. So we've seen it now 2 or 3 separate times in the US where we've seen this bird something to cow transmission events. Once it gets into cows, we think it moves at least within the herd through the process of milking those cows. So contaminated, because again, remembering this virus in cows is replicating in the udder of the cows. And so through the milking process, that probably, moves it from one cow to the next. And then we have cows again, moving from one farm to the next. And that is at least primarily how we think the virus is getting around.
What is the status or plan for vaccination against HIV-N1?
So one, we can talk about vaccinating the cows, right? And this is it's a viable option moving forward to try and perhaps help control the outbreak is to vaccinate cows. There are at least a couple of products that are in fairly advanced stages of approval by the USDA. In my understanding, with all things going well, it shouldn't be too long before some of these vaccines are actually approved. That doesn't mean they'll be used. It just means that they've met all the criteria for being approved. From a human perspective, if there's one of these emerging pathogens that we're most prepared to respond to with a vaccine, it probably is H5, which is a good thing. We've been dealing with this virus, the royal we, for 25 years now. So there have been many clinical trials with H5 vaccines, not necessarily against the current version of it, but from some of their ancestral viruses. So we know how to make vaccines that at least induce a good immune response against H5. The US government has stockpiled some vaccine, meaning they do have so many millions of doses of H5 vaccines. Again, not exactly against the virus that's circulating, but all of the evidence we have suggests that they probably would be protective. So from that point of view, there are vaccines again available for the human side of things. And of course, there's not enough for everybody who's going to want the vaccine. So, but at least we know how to make it. We know the process to go through.
What do you think is the best way for society to avoid having a pandemic from H5N1?
I can certainly tell you what we would want to do to stop the pandemic. I can't, unfortunately, necessarily tell you how to get there. But what we've clearly got to do is remove or reduce as much as we can the human exposure to this virus. So again, let's think of this maybe a numbers game, a roll of the dice. Right now, the H5N1 bird flu virus is still very, very much a bird virus. Right? Really poorly infectious for humans, unless there's some specific situations and exposures going on. And so again, it's a numbers game, but the virus likes to change. The more opportunity it gets to even replicate a little bit in humans, the more chances are it's going to pick up some of these key changes that make it more infectious for humans. So what we absolutely have to do is do everything we can to reduce the number of people exposed, and that's through controlling the virus in animal populations. Thinking wild birds, that's really, really hard to do, right? Theoretically, you could think about vaccinating those, but practically there's no way for us to do that right now. But we can do things about it in more the domestic poultry and cattle, dairy cattle populations, whether that be through vaccination, whether it be through more research to understand how the virus is moving around. And with that information, we can do things more about stopping it. But that's what we got to get to. We can't eradicate this virus from everywhere. That's just not.
And what would you like to continue learning about H5N1 or other viruses? Are there other things you would like to study?
We can take an influenza virus from, pick your favorite animal host, a dairy cow, a sea lion, a dolphin, a fox, and we can, with some degree of confidence, tell you, well, that virus probably is not going to infect humans very well. What we got to get to is, of those that aren't infectious for humans now, which of those are actually more likely to be able to make the leap to humans? We're not good at that. I think with some of the emerging technologies and some of the more sophisticated sort of sequencing evolutionary type analyses, perhaps with the more machine learning AI type approaches, that's where I think we can gather some of the information we have and become a little bit more predictive. So it's not just yes, this virus can infect humans, or no, it's of the no's, these are the real ones we have to watch out for.
And do you think that the avian viruses are the most likely or something else?
The most likely are probably viruses in swine. So, why is that? Well, one, if you take a look at the viruses that are in swine, they had many, many more of these sort of human virus properties. So they're more human virus-like than they are avian virus-like. I think one of the saving graces in terms of why we don't get more pandemics from swine is there's actually quite a bit of similarity between the influenza A viruses in swine and the influenza A viruses in humans. So we have quite a bit of immunity to most of those swine viruses. But over time that will change. And I think the 2009 pandemic was a great example of that. That even at that time we were really, really worried about H5. And where did the pandemic came from? It came out of a swine somewhere probably in North America.
When most viruses have reached this point where we are now with H5N1, do they normally then become humanly transmissible?
I would say not necessarily. So to be honest, we haven't really seen what we're seeing now with necessary and avian influenza virus. Typically, we talked about wild aquatic birds being the main sort of reservoir for influenza A viruses. And that's, where of all these different shapes and flavors of influenza viruses, that's where most of them live. And most of them don't actually move out of that host. So the little exposure to humans. There are some other viruses. There's another virus we keep a really close eye on, a so-called H9N2 virus, that has moved out of sort of the wild bird into poultry through many, many regions of the world. But it's not in the Americas. That hasn't been infecting cows. It hasn't been infecting foxes. So, I think we're in uncharted territory a little bit right now with the H5N1. So to say we've been here before and haven't seen a pandemic, we can't say that. But we also can't say we've been here before and have seen a pandemic. So it's all new.
It was so interesting to speak with you, Dr. Webby. Thank you so much for your time.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.